and it's got a few wires and jazzy things coming out the front of it.
What's one thing that people might find surprising or mind-blowing about this work?
Not only can we wirelessly record from the brain of a moving, living creature, but we can control it just by shining a light into its brain.
Hello, and welcome to Technology Network's latest video series, Science Spotlight. Today we're hearing from Stuart Greenhill. He is absolutely fantastic, and he's here to share his exciting research. I know you're going to love it, so let's dive on in. Hi, Stuart. How are you doing?
I'm doing very well, thank you. How are you?
Really well, thank you. So, can you tell us a little bit about yourself and the research you've been doing?
Oh, yeah, of course. So, my name's Stuart Greenhill. I am a neuroscientist and a pharmacologist here at Aston University in the middle of Birmingham, UK. What we do here in my lab and in my collaborators' labs is that we're interested in how the brain develops, how the brain's areas talk to each other, and how that can go wrong in conditions like epilepsy and schizophrenia and other such related brain disorders, where maybe the circuits in our brain are forming a little bit differently. The bits of our brain may not be talking to each other quite the same way. What we're interested in is seeing how we can intervene in some of that circuit level activity and maybe come up with some new drugs or some new treatment avenues to make people's lives a little bit better who have those conditions. The way we do that is by looking at the electrical activity of brain cells. We call this electrophysiology. And out in the lab, we can do that on an individual cell level where we can record with little tiny needles from individual brain cells. We can record from slices of brain that we can get from animal models of disease or that we can get from human patients who have undergone surgery at the children's hospital across the road. By looking at all that information, we can build up a picture of how our brain cells are supposed to talk to each other and how that goes wrong in certain conditions. Now, what we also do is develop a series of animal models. It's often difficult to use animals for research. We don't really want to have to use animals for research. But in some of the more complicated conditions, we still don't understand enough about the brain's activity to be able to simulate it in a dish or to be able to simulate it in a computer.
Hmm.
So what we do is we try and come up with better ways of recapitulating the disease states that we're interested in, and then putting them into animal models and using animal models in a kind of nicer, less impactful, less harmful way, so that we can get more data out of fewer animals, and that so that those animals can have an easier time within the experimental model. We don't want to do too many harmful things to them. So what we've been doing is working with a company called AD Instruments to develop and deploy what we call a telemeter. And that's just a little device like this. And we can implant it inside a full-grown rat, and it can just sit under its skin in its tummy. And it's got a few wires and jazzy things coming out the front of it. Now, these are a collection of recording wires, so we can record the electrical activity of quite a large part of its brain. But also we've got this blue fiber here, which is connected to a very powerful LED inside the telemeter. And that means that we can use a special molecular biology construct that we put into the brain to control the circuitry of the brain in certain conditions with this light. And we can do that while the rat is running around and living its best life
Hmm.
and becoming epileptic through the different models that we use, for example. So we
Hmm.
can follow through the electrical activity of the rat's brain as it becomes epileptic, and then use this little optical fiber to intervene in that activity and try and see the limits of that circuit function within normal brain function and within the disease state that we're interested in.
Incredible.
So, what I'm going to talk to you today about is a piece of research that we've been doing in collaboration with our industrial partners that ended up as a poster that we've taken to a couple of major conferences over the last year or so. We've gone to SFN in America, we've gone to FENS in Europe, and we're currently in preprint stage for a publication that should hopefully be coming to a peer-reviewed journal pretty soon. What it is the culmination of this product development, where we've taken the knowledge that the company's got and the knowledge that we've got and combined them using something called a knowledge transfer partnership. Now, this is a really good scheme run by the British government, whereby we get a little bit of funding money from the UKRI, UK Research Councils, And between us and the company, we worked for 2 years testing the product, refining it, working out all the little niggles and bumps in it, and figuring out effectively the best way to use it so that we get really good early access to this excellent piece of kit. And the company gets a lot of our experience so that they can use it to help other people who might be using the kit in the future. So it works really well for us, and we've had quite a successful time out of it, and we're really chuffed to be part of this partnership. So here's the poster in its entirety. There's quite a lot to it. I'm not going to go through every single little bit of text and stuff like that. But what I am going to do is just take you through a few of the major kind of figures in it, and try and pick out some of the highlights of what we've been doing. So as I say, our whole animus here is to get more and better data about better animal models of quite difficult to treat conditions in a way that doesn't use too many animals and in a way that doesn't make the animals that we're using suffer more than they need to. It's something that we call in Britain the 3 Rs: replacement, reduction, and refinement. We want to use as few animals as possible. We want to replace them where we can. We want to refine what's being done to those animals in the best way possible, because effectively, stressed animals leads to bad data. If I want to record how a brain functions, the way my brain functions when I'm really stressed out, it's very different to the way that my brain functions if I were lying on a beach sipping a pina colada and reading a nice book. So we need to keep these guys as happy and as normal and as stress-free as possible throughout their experimental lives so we can gather the right data, not just any old wiggly line. We want to know exactly how their brain's functioning on an electrical level. So we know that we're not there yet with neuroscience. Science doesn't know everything. Otherwise, it would stop, as somebody once said. So we need to be able to use whole brains. We need to use bits of real brains in some cases to see how these disease states arise, to see how these really complicated circuits all talk to each other and make the functions of our brains at quite a high level. Now, the more data we can get from a brain and the longer that we can record from that brain while its conditions change and while the animal develops or while it develops a particular disease, the better understanding we get because we can compare the function of that animal's brain before and after we've done some intervention. And that might be setting up a disease model. We might be able to trigger an epileptic seizure, for example, or we might be treating it with a drug. We might be treating it with a new treatment, and seeing how it responds during that treatment before and after. So it's a within animal control, and it's much richer, more usable, more repeatable data. And so by using this little implant, we're able to do that for quite a long time and at quite a high level of detail. So here's a rendering of how it looks within the body of the rat. You see the little device I showed you earlier lives down here in the abdomen. And we can implant it inside the body. It's all watertight and it's all biocompatible. It doesn't have any inflammation or any nasty effects to the insides of the rat's abdomen. And what happens is we've got the EEG wires, the electroencephalogram wires, that run all the way up to the head, and we can attach them to little screws that we put into the brain area that we're interested in. So we might be interested in the somatosensory cortex or the motor cortex. We might put a little electrode all the way down into its hippocampus, where its memory and navigation and things like that takes place. Regardless, we will connect these wires up to the screws over the brain area that we're interested in and record an EEG. You know, you might have seen or may have had yourself the little wires that you can stick on people's heads to record their brain activity. That's exactly what we're doing, but in a little implant that lives inside the rat all the time. And then wherever we want to put this little optic fiber to zhizh up the brain to our particular demands with this special protein that we use to control the brain cells, we can implant that fiber as well. And between us as a team, we came up with a mechanism to implant this stuff so it's completely subcutaneous. It's all under the skin. There's no extra little head stages or visible equipment on top of the rat. It's all contained within the rat. And that's quite important because this rat over here on the left, he's one of our fellas that's had the full implant, right? And if you look at him, he doesn't really look any different to your average rat that, well, not quite what you get off the street because they tend to be big and brown, but your average rat that you would get in a lab. He looks pretty much bang normal. So if we're doing a really complicated behavioral experiment that requires multiple rats to interact with each other, and we want to see how social they are, we want to see how they deal with each other, how they interact on a very kind of subtle level, if we're looking at something like schizophrenia or autism or something that has a sort of cognitive component to it, we don't want Jeff the rat to come in with a big thing on his head and make himself obviously different from the rest of the crew. We want our experimental rat to look, smell, act exactly the same. So he can't be limping around, he can't be in pain, he can't be all puffball, because when they get stressed, all their hair goes up on end and they look and act very, very different. So this guy just tonks around as if there was nothing different about him whatsoever. And that's a real advantage. Moreover, we can record from him and use him in our experimental runs for ages and ages and ages, because these telemeters are wirelessly charged. They sit on a great big pad like you put your phone on to charge it, and they're constantly being topped up in terms of their power level, which means we can run them at a really high level of detail, really, really high level of data acquisition, and do all this jazzy optical stimulus to entrain the brain to whatever frequency we want so we can interrogate its circuit function. So here comes some wiggly lines and some graphs, right? And this is the world in which we live. This is what we want to look at in terms of the brain function. To us, it's all about the electrical activity of a cell, of a synapse, of a circuit, of a brain. And that comes down basically to a bunch of electrical signals. And if you look here at the top left, this is the raw, unfiltered EEG, the electroencephalogram. It's the combined electrical activity of this area of the rat's brain. And what we can do is, using a filter, just by filtering it mathematically and cutting out different frequencies and looking at it individually, is break it down to the different kind of major component waves that we see in various bits of our brain and in various functions of our brain. So down the bottom, we've got the gamma wave. The gamma wave is your concentration wave. You see it when you are thinking about stuff, when you're paying attention to things. It's also your dream wave. When you have rapid eye movement sleep and you're dreaming about stuff, your brain flips into this gamma-like state, and it's where we dream in this gamma. We've also got things like alpha and beta, which have specific functions in things like movement and vision and all that kind of stuff. And then delta and theta, which tend to be more kind of long distance and coordinating waves. The slower your brain oscillates, generally, the more of it kind of links up and talks to each other. So again, if we go back to being asleep, before you start dreaming, the brain waves get slower and slower and slower and more global until your whole brain is oscillating up and down like the ocean, and it's very relaxing. And it snaps into this fast gamma when you start dreaming. And so by looking at the interactions of all these little brain waves, we can figure out how the bits of the brain, the individual pieces of cortex, the individual areas of your brain, are talking to each other and influencing each other, because one wave tends to modulate the other. So the amplitude of a wave often obeys the amplitude of another wave, and they sit on top of each other in something we call phase amplitude coupling. And that's how our brain all synchronizes up, and we know what, literally one hand knows what the other is doing. That can go wrong. That can go wrong in things like schizophrenia. If we start to slip the coordination of these oscillations, then we can start to experience things like deja vu. We can start to hallucinate. We can start to perceive the world differently to what it might actually be. And so the more we understand how these waves talk to each other and interact with each other, the better understanding we've got of what people are going through if they're experiencing some of these conditions. Now, over on the right-hand side, we can see some graphs, and these are called fast Fourier transforms, right? We can break down this raw wiggly line into lots and lots of frequency components and look at how strong, how powerful an individual frequency component is within that complicated wave. And you can see over here, if you look at this green dotted line, for example, if we turn on our little stimulator and we make our jazzy proteins dance to a 10 hertz wave, we can entrain the brain to also fire at 10 hertz. So we can basically get our rat's brain to obey the signals that we're putting in. Now, that's not just for a party trick. We might want to boost, or in some cases, diminish the power of some of these waves. We can look at it a little bit like your noise cancelling headphones work. You know, you've got the little microphone that records the noise outside, and then if you flip the phase of that, it all cancels each other's out, because the peaks cancel the troughs out and you just get a flat line. We can do that with these waves. We can make them disappear by playing them back to each other out of phase, for example, or we can make them much bigger by playing them in phase and basically kind of get the brain to do what we want it to do in a given experimental condition. And so we can do that on the top one, and that's the rat that we showed you before, moving around, quite happy, charging up all the time. And then underneath, we've got ones where we've taken the optical fiber and we've changed it. We've connectorized it, we've put little extensions on it, and we've basically messed around with its configuration to push it as hard as we can and see what happens. And it all seems to be the same. So we could take it away, refurbish it, reconnect it afterwards, and still use the implant again. Because we wanted to test it in as many ways as possible to make sure that whatever I wanted to do with it or whoever bought it wanted to do with it, they would be able to achieve that and get the same kind of repeatable results. And you can see still, if we look at the little dotted lines, the brain will do what we tell it to do. Sometimes actually the brain does what we tell it to do and a little bit more. If you look at the 10 hertz ones, you've got a kind of bump at 10 hertz and a bump at 20 hertz. And that's because we'll get harmonics. So the circuits that we are in training, they will do things sometimes at half the frequency or double the frequency that we're telling them to do because they've got their own intrinsic activity that they want to try and keep to. But still, we can make them do roughly the frequency that we're interested in getting them to oscillate at. And so this is just a bit of stats that shows over a period of time, we can have these rats recorded from, and even after 3 months, 4 months, we can still get them to entrain their brain to the frequency that we want them to do. So we can take these implants, we can record from really high fidelity brain signals, we can control those brain signals with little bits of light that are coming from the implant. We can keep these rats going for months and months and months and look at them before and after we've triggered their epilepsy, before and after we've done their drug treatments. We can do all kinds of behavioral experiments for them. And instead of having to use 60 rats, we can use 6 rats, because we don't have to do loads at different time points. We don't have to do loads and loads and loads of repeated experiments. We can use one rat to record for months and months and months and get as much data out of that rat's brain as possible. And I think we've got a kind of moral imperative to do that. We can't just go around doing things the way everybody's always done it and using loads and loads of animals in the process. We've got to try and do our best to reduce the amount of animals that we use, but not only that, to get better, more usable, more what we call translational data out of those animals. And translational data is something that's going to find its way as quickly as we can into a clinic, because we're not doing this to cure rats from epilepsy. I mean, that would be nice, but ultimately we want to help people who have got epilepsy out there in the world. So what have we done? We've refined this product, we've developed this product, we've worked with the company, and we've tested it in a whole multiple range of ways. And we think we've come up with a pretty robust way to use it, and a way that you wouldn't even be able to tell that it was implanted by putting things in at specific angles, by doing things in specific ways. These rats just look like normal rats. The good thing is we've come up with a way to do it relatively straightforwardly, so that you don't need a load of specialized knowledge. You don't need to build your own little brain implant rigs or anything like that. You can go and buy this stuff off the shelf, learn how to do the surgery, and off you pop. So it's democratizing it, if you like. It's opening up this kind of research to more labs across the world, because they might have some really good ideas, they might have some really good models, they might have all the brains in the world, but they might not have the money or the technical ability to do certain things. But if we can do it like this, then that allows them, that frees them up to do that research. And it frees them up to do that research in a really nice longitudinal way.
That was incredible. Thank you so much. So, I do have a couple of questions I really want to ask after having heard everything. So, given the advancements in alternative methods, what sort of role do you think that animal studies still play in this type of research?
So we actually, as a sort of scientific endeavor, have made in the last couple of decades massive strides in a lot of places. One of those places is in things like toxicology testing. So if you want to do safety testing for drugs or chemicals or all that kind of stuff, you can literally recapitulate a liver or a kidney or a pancreas or something like that in a little microfluidic system. It's just a little bit of plastic full of
Mhm.
cells. And that's brilliant. That means that you don't have to give it to a rabbit or a dog or anything like that they had to do back in the 80s and the 90s. It really has moved on loads and loads. And so the use of animals in all those kind of areas is going off a cliff, and rightly so.
Mhm.
And the guys there are really embracing that kind of alternative technology and running with it and coming up with really good results, because it makes it faster. and it
Hmm.
makes it cheaper. Um, and it's just, morally more defensible. However, if we're talking about how fundamentally we work,
Hmm.
we know how the simple bits of us work in terms of how the tissues are all put together and how we might release a hormone here that'll end up there. But still, on a fundamental level, we don't really know what's going on a lot of the time inside one of those cells, all the messengers and the proteins that are all interacting with each other. We're fortunate enough to live in the post-genomic era, so at
Hmm.
least we know how many proteins there are.
Hmm.
But every time we discover something, it's a little bit like the particle physicist guys, they're all smashing things together, and then a whole heap of other stuff comes out, and they're like,
Yeah.
wow, we're going to need a bigger particle accelerator.
Yeah.
So we're basically at that kind of stage. We know that these proteins exist, but now we've figured out a whole bunch of stuff. That there's much more layers of complexity underneath them. And in fact, the Nobel Prize was on a couple of months ago by the guys that figured out that there was this microRNA that was sort of doing all this sort of extra stuff that no one
Hmm.
had ever considered before. So it used to
Yeah.
be we thought we had the blueprint, but now we've got a blueprint and some mad dude with scissors all chopping it up and rearranging it whichever way he seems to want.
Hmm.
And that's just in a cell, right? And it could just be in the most
Yeah.
basic possible imaginable cell. So if you extrapolate that to a neuroscience, and you extrapolate that to the function of a neuron, a brain
Hmm.
cell, which not only is a complicated cell in its own right, but sort of processes information, makes decisions, has a threshold. It's like a little tiny computer in
Mm.
and of itself, a little analog computer. And yeah, we're very clever and we sort of understand how a neuron works, but not really. But then we take 86 billion neurons and put them all together, and then it gets quite complicated. And so we're not entirely sure how most of the circuits in our brain are really, really working. We're getting there, we're getting that understanding.
Hmm.
But if we don't know what the right answer is, it's very difficult to figure out when it goes wrong.
Hmm.
So what we have to do is still on certain levels, if we want to look at certain conditions, get whole brains or slices of brains or large collections of neurons and see what they're doing. And we need to see what the neurons do, not what the neurons can do. You can
Yeah.
make a neuron do anything. You can make it glow in the dark, a flash, or set at any voltage you want it to.
Mm.
That's lovely, but we need to see what they're doing on a circuit level sort of naturally and as naturally as possible. And because we can't build that from scratch yet, yes, we can build parts of it from scratch. We can use organoids for different parts of the research.
Hmm.
But in some cases, we really need to use that natural brain circuitry to
Hmm.
interrogate what's going on. And in those cases, we either need to use human brains that are taken out of surgery. So if you've got really intractable epilepsy, you might have the little bit of your brain that's epileptic removed,
Hmm.
and we can take it over to the lab and slice it up and poke it with electrodes. We ask permission, of course. We're not just doing this,
Yeah.
randomly. Um, but so we want to poke it with electrodes and want to record the electrical activity, and that's wonderful. But it's very limited in terms of how much tissue you get. You can't, demand a particular piece of tissue. It's all about the
Hmm.
need of the patient, isn't it?
Yeah,
So
absolutely.
if we want to interrogate a particular part of the brain and we want to interrogate how a particular process happens, then we kind of need to use
Hmm.
something like a rat in order
Yeah.
to be able to do that. And we use rats because they've got a complicated enough brain that we can extrapolate it to a human, but they're not so complicated that they'd be like a monkey or something
Yeah.
like that. So we want to use it's a bit reductive to say that there's a hierarchy of species, but we want to use the kind of smallest, least complicated brain that we can
Hmm.
to get the information out of it.
No, that makes a lot of sense. Thank you. So, my second question is, how does this kind of work improve studies on brain activity in rodents compared to kind of other tools? I know we've kind of just touched on this, but I'm also wondering, could it be adapted to use in mice and other kind of animals too?
Yeah, so there are versions of it that are used in mice. They tend to be smaller. They don't have the optogenetics in it, but they do make one that is small enough to be fitted into a mouse. You don't have to record from a brain.
Hmm.
It'll record any electrical potential in the body, so you could record from the
Okay.
heart or the gut or anywhere where electrical signals are used to coordinate activity.
I hadn't thought of that. Yeah, cool.
Yeah. So how it helps and science is always sort of telling everybody else that you're doing it the best way and that their way is rubbish, and that's not true. There's usually a benefit to any which way of doing
Hmm.
a scientific experiment as long as it's done robustly. But this opens up a new um, accessible avenue for people to do what we call longitudinal studies.
Hmm.
So if we take a telemeter that isn't charging wirelessly, that's got quite a limited battery life, and you might be able to use it for a week or a month or whatever,
Hmm.
and that's grand. But then, of course, it's over. It runs out of battery. You can't get any more data. Because this is wireless, because it's charging, because it's high powered and all of that stuff, we can do these quite high level and quite complicated experiments for a long period of time and get loads of developmental data and loads of sort of disease progression data out of these animals. So, for example, if we take our epilepsy model, it takes about 8 to 10 weeks for the epilepsy to appear.
Oh, okay.
What we do is we'll give a rat some drugs to give it an epileptic seizure,
Hmm.
and then we'll stop that epileptic seizure so it's just enough to trigger epilepsy and without causing brain damage, for
Hmm.
example. And then nothing happens. Everything goes very quiet in the brain and it behaves quite normally. And
Hmm.
then suddenly, over the course of a couple of months, you start to see this activity coming, and the behavior of the rat will change. The appearance and behavior of its cells will change. And then suddenly, it will develop spontaneous recurrent epileptic seizures. But that takes a
Oh.
couple of months.
Hmm.
So if we wanted to record that activity throughout that entire time, that's 2 or 3 months worth of telemetry that
Yeah.
we need.
Hmm.
Furthermore, if we then wanted to wait until that rat had become epileptic and then give it a bunch of drugs to see if we can treat its epilepsy, that's another couple of months on top of that.
Yeah. Hmm.
So that's a long old experiment. And so in the past, we had options either to just do the telemetry for a little bit in a
Hmm.
targeted fashion, or to turn it off and on again to try and preserve the, when you got your phone and you're in the middle of nowhere and you keep turning it off
On
to
and
try and
off.
get the battery. Yeah.
Yeah.
So so we could do it like that and just turn it on and wave it about every now and again. I guess a couple of days to see what's going on.
Uh-huh.
But this allows us to record 24/7 throughout the whole experiment and get all that data. And not only that, it records at a higher bandwidth, so we can get higher frequencies.
Hmm.
So I'm sure you're all very familiar with the Shannon-Nyquist theorem of information transfer. But for those that aren't, if we are sampling something digitally, if you like, if we're taking little samples of a wave,
Hmm.
we have to at least, at the very least, sample that wave at twice its frequency. Otherwise, we just get a flat line or a wonky kind of aliased thing. If we want to see the actual shape of this wave, we need to oversample it
Hmm.
and sample it much more times than that wave would oscillate in a given second. Now, if we're limited to sampling at, say, 256 hertz, we can only look really at activity up to about 100, 120 hertz.
Okay.
If we are using this fella, we can sample at 2 kilohertz. And so we can look at pretty much every bit of activity the brain can possibly do, because most of the time it's in the tens of hertz. But sometimes our brain oscillates really, really fast. These very high frequency oscillations, and you can only see them if you're recording at a really high bandwidth. And so
Oh,
because we can do that, we can pick up stuff that we might have never been able to see before in other experimental preparations.
nice. Cool. So my third question for you is that your poster specifically mentions sort of improved tolerability and reduced risk for the animals, and I know that's what we've been talking on as well. But how do you see these kind of ethical and practical advances influencing broader adoption of the technology and, neuroscience research in general?
I think, the science is funded by the public in general.
Hmm.
Um, and we have to be responsive to the attitudes and the opinions of the public, because if we're getting paid for by taxpayers' money, we need to
Hmm.
be cognizant of what those guys want, really. And so it's not only a sort of isolated moral imperative for us to do things better with animals just because it's the right thing to do.
Hmm.
The people that pay the bills want that as well.
Yeah.
Everybody, people in Britain, people in Europe, people across the world generally want animals to be treated nicer, whether that's in farming, whether
Hmm.
that's in scientific research, whether that's in, banning various practices that people,
Yeah.
think are perhaps outdated in various parts of society. And that's just, progress, isn't it?
Yeah.
We have to do better.
Hmm.
And so I think, as well as the kind of regulatory imperative to do that, there's a financial one as well,
Hmm.
because the the more we can get out of fewer animals and the quicker we can get it, the more cost effective that research will be. And in a time where, there's not a lot of money sloshing around, it's just makes sense to be able to uptake that kind of technique and do research more effectively in a whole bunch of ways, right? Whether it's
Hmm.
whether it's sort of isolated philosophical need to do things better for the animal's sake, whether it's simple pragmatism because we've got less money, or
Hmm.
whether it's because we can actually get more done in a finite length of time. We can develop this stuff and then move on to the next thing.
Yeah.
It all just makes sense
Hmm.
intrinsically to me.
Perfect. Well, thank you so, so much for sharing this research with us. It's been brilliant speaking with you. And if anyone does want to get in touch with you and chat about your poster or your work, what's the best way that they can do that?
If you just stick my name into the search engine of your choice, obviously you'll get the university
Hmm.
webpage. Usually the best way is just to drop me an email,
Hmm.
s.greenhill@aston.ac.uk, or just to hit me up on anywhere you might find me. We publish papers, we go to conferences. If you see us around, then just give us a shout, and we're always interested in collaborations or talking about our research.
Perfect. Well, thank you so much, Stuart. It's been brilliant.
Thank you.
Thanks so much for watching this Science Spotlight episode, and I hope you'll like and subscribe, and I hope you'll come back and see us again very soon.